Welcome to Grand Rounds. It's my pleasure to introduce Jeffrey Glassberg. He is a tenured professor of emergency medicine, and hematology and medical oncology at the Icahn School of Medicine at Mount Sinai, and the director of the Mount Sinai Center for Sickle Cell Disease. Dr. Glassberg is the first emergency physician to run a sickle cell program where he practices in a unique way, seeing patients in the emergency department, in the hospital, and in the sickle cell clinic. He has grown the Sinai sickle cell program into one of the largest and most successful in the world, with nearly 1,500 patients and more than $25 million in NIH funding. Mount Sinai's program is also one of the largest providers of access to cutting-edge treatments for sickle cell disease, including monoclonal antibodies and gene therapy cures. To mitigate the critical shortage of expert sickle cell physicians, Dr. Glassberg created the Mount Sinai Sickle Cell Fellowship, one of only 2 dedicated sickle cell fellowships in the United States. Dr. Glassberg's research spans the translational spectrum, including pulmonary complications of sickle cell disease, novel biomarkers for vaso-occlusive crises, novel gene therapy targets, and agents for the prevention and treatment of vaso-occlusion. Please join me in welcoming Dr. Glassberg to Grand Rounds.
Okay, thank you very much for having me. I will stay close to the microphone. This lecture goes through 3 of my favorite projects that are going on right now, and I've framed it around a sort of past, present, and future in sickle cell disease. And I want to finish early, so I'm going to, there's my consulting and disclosures. I'm going to buzz through the background about sickle cell disease and assume that we don't need to cover board review type of stuff. So this is about sickle cell disease and hemoglobin and the gene mutation that people have, fetal hemoglobin. I'm skipping all of it. This is about what sickle cell disease is and how it was the first genetic disease discovered as a single mutation with a single incorrect letter, and how that gene gets translated into RNA, transcribed into RNA, and then translated into a protein, et cetera, et cetera. We have these different genotypes. Even though everybody has the same family of mutations, we see a lot of diversity in how sick people are. And that has to do with these other factors beyond sickle cell disease. But the bottom line is we see manifestations in every organ system because sickle cell disease is a disease of the blood, and the blood feeds the whole body. Right now, we think the life expectancy is around 50. It's a little hard to tell because some of the best studies were done before hydroxyurea was widely in use for young people, but 50 is a kind of useful estimate to benchmark where we think the life expectancy is. Okay, so I'm going to go through sort of how things were in the past and how we're trying to rectify that with one of our research projects. So let's talk about hydroxyurea. Hydroxyurea is a drug that is approved for the use and treatment of people with sickle cell disease. Basically, it has 3 mechanisms of actions. It raises your fetal hemoglobin levels. It reduces your leukocyte count, all forms of leukocytes, and it also has a nitric oxide on the side, so it's a nitric oxide donor, which helps with vasodilation. In this New England Journal of Medicine article in 1995, it was shown to reduce the frequency of pain crises. Three years after that New England Journal article, the drug was FDA approved. And 10 years after that, it was still basically being used as a drug only for people who met the criteria in that New England Journal article, which was people who had a lot of pain crises. So even though dozens and dozens of articles had been published showing that this is a drug that actually improves organ function, mitigates the complications of the disease, doctors were still using it basically for people who had a lot of pain, which didn't make much sense. And I was, at this point, a resident in training, and it never made that much sense to me because it's a drug that treats the pathophysiology of the disease. Not everybody with sickle cell disease has frequent pain crises. Some extremely sick individuals have very few pain crises, so it didn't make sense to me that we weren't using it for everyone. By 2008, there were consensus conferences reviewing all this literature, and still the community didn't really come down on the side of we need to use this drug for a broader population. At this point, now this is, we're talking about data from 2017, 20 years after the FDA approval, and we're still going back and forth about who we should be using this drug. But by this point in my clinic, when patients ask me if they should be taking hydroxyurea, and they would say things like, well, I'm not sure I need it, and there's this stuff on the side effects, and I would say to them, this is actually a pretty simple decision to make. If you want to live longer, you should take the medicine, and if you don't, you shouldn't. Any questions about, well, I don't have a lot of pain crisis, or I'm not sure I want to take a pill, or what if I have a side effect? Take the medicine if you want to live longer. If you have a side effect or an issue, then you can reevaluate, but we know that this drug will make you live longer, so if that's something you'd like, then you should take this medicine. So this issue of having taken, the drug was really discovered in 1977, I believe, but having taken 40 to 50 years to figure out that a medicine makes people live longer, to me was really not ideal. This is Bernard Herrick, James Herrick, the hematologist who discovered sickle cell disease in 1909. And so, we were, I felt like sort of innovation was really stagnant in sickle cell disease. We were taking too long to figure out which drugs worked. There was a lot of research, and this is actually my own research, where we were pulling drugs off the shelf and seeing, well, maybe this will help. And so this was our trial of inhaled steroids, which we did a small pilot study, and we showed that people who took inhaled steroids who do not have asthma did better. Their biomarkers improved, so we were very excited about this. And that led to an R01. We did a trial, 80 patients who were randomized. They took inhaled steroids for a whole year, and in the end, it basically looks like inhaled steroids are worse. So I wasn't super hopeful anyway. The idea that some asthma medicine that we had pulled off the shelf is going to transform the landscape of sickle cell treatment was really, I knew that was never going to be the truth, but this was just another one of many efforts to find new treatments that didn't work. But then at this time, we were having a lot of pharmaceutical companies that were developing agents for sickle cell disease. And by 2018, we had 3 more FDA approved. They work by all different mechanisms. Some are anti-sickling, some are anti-adhesive, some are antioxidant. So we had all these new medicines. One of them is crizanlizumab. That is a P-selectin blocker. You take it once a month. And you can see here on the right, the Kaplan-Meier or sort of hazard ratio curves where people who get the high dose medicine have longer time to first pain crisis. Basically, individuals who took this medicine have about half the number of pain crises with sickle cell disease. So this was an exciting drug that got FDA approved, I believe, in 2017 or 2018. There's voxelotor, which also came to market. This is a small molecule. It's an anti-sickling agent. You take it and basically kind of gets in there with your hemoglobin inside the cell, and prevents it from sickling by raising the oxygen affinity and also just sort of getting in the way of the hemoglobin sticking together inside the cell. And this is a sort of cartoon depiction of what the molecule does, and then you have happy biconcave red blood cells. Patients who take this medicine, since they're not sickling, they're not hemolyzing, and they have higher hemoglobins, and you can see these waterfall plots that patients who took the highest dose had the largest hemoglobin response. We've had pretty good responses with this drug as well. We have a lot of patients on both of them. Sometimes you can have a patient go from 7 to 11 or 12 hemoglobin taking this drug. And also very important, people did not have more pain. They had slightly fewer vaso-occlusive crises on voxelotor. So now we have this new problem where we have all these drugs. When a drug gets FDA approved, it does not get compared to other newly approved drugs. It gets compared to the standard of care. So hydroxyurea was in some of these trials, but there was no comparison of the drugs together. And furthermore, there was absolutely no usage of these drugs in combination. So while they're all FDA approved for sickle cell disease, there is no precedent for how to use them together. Other than some basic like drug-drug interaction. Do we have a CYP450 issue or something like that? So we have a new problem, and I certainly didn't want things to go on for 40 years while we're trying, the way we did with hydroxyurea, taking that long to figure out that the drug, because it treats sickle cell disease, makes people live longer. So looking at the inclusion criteria from some of these trials, I basically saw that the problem happening over again, right? The clinical trial that led to FDA approval of crizanlizumab required that you had to have a certain number of pain crises. And what happens when you do that with your New England Journal trial? Well, when the drug gets FDA approved, even though it's approved for sickle cell disease, all the insurance companies basically have their pharmacists pull the New England Journal study and write a coverage policy that's based on the inclusion criteria from that article. So you're already headed towards a situation where only people who have a lot of pain, which is not everybody with sickle cell disease, is going to be able to access the drug. So that is what led me to write this grant, which was called Real Answers, and it's based on leveraging a large registry consortium that we were in that has these 8 sites, 2,400 patients in the registry, and we've got all these very nice validated data elements and PROs and labs and all kinds of stuff. So I said, well, maybe we can sort of modify this multi-center registry to something that actually can figure out how to use drugs together and which drugs are better. So we made sure we're collecting the outcomes from these clinical trials, like hospitalizations, et cetera. But also, I wanted to look at the outcomes that matter to us as clinicians. And what is it that drives survival in sickle cell? One of them is organ injury. So we think about blood as an organ, blood injury, we call that hemolysis. So we're going to look at hemolysis, and certainly hemolysis has been correlated very, very strongly with survival in sickle cell disease. So we have ways of dealing with the fact that markers of hemolysis have a lot of collinearity, and we basically distill them into one score. ProBNP is another biomarker we follow in sickle cell disease. It is strongly correlated with mortality, regardless of whether or not you have a lot of sickle cell pain. And then another really nice biomarker we use in sickle cell disease, a marker of kidney injury, is how much protein you're spilling in your urine. And one of the really nice things about these biomarkers, like hemolysis, protein spilling, and proBNP, is they're disease sensitive, they're not cumulative, right? So creatinine is a little different than protein spilling, because creatinine has somewhat of a cumulative nature to it, that over time your creatinine will go up, can go up and down with acute issues as well. But in general, until you're at end stage, your microalbumin to creatinine ratio is a very disease-sensitive biomarker. If you put someone on a drug and they're spilling protein at 200, it might go down to 100. And so these are biomarkers that we felt we could use to assess the effect of these new drugs on organ injury. And we like that they're sort of distal biomarkers, like the selectin inhibitor. We actually see protein spilling get better, but it's not like a drug that's affecting the kidney. What you're seeing is the disease is getting better and the organ stress is improving. So we're not going to conduct a randomized controlled trial for every possible combination of these drugs. In fact, if you go back to your combinatorics, which my kids are learning in school, you've got 4 drugs. There are 15 potential ways you could combine those 4 drugs. And then if you wanted to compare each of those head-to-head in a randomized controlled trial, combo 1 versus combo 2, combo 1 versus combo 3, you'd be doing clinical trials for about 150 years. And this is a rare disease, so it's a non-starter. This is not going to happen. And yes, RCTs are better, the evidence is high quality, but it's a fantasy if you think that you can do randomized controlled trials to figure out which of these drug combos are the best. So what do you do in that situation? Well, you can use something called target trial emulation. And so what we're doing here is we're taking this registry framework of these 8 centers, thousands of patients, and we're collecting the same data that you would collect if these patients were in a clinical trial. You would get regular labs, your adverse events would be adjudicated, every hospitalization would be recorded, and then what medications you are on, all your concomitant medications, especially the disease-modifying sickle cell medicines, would be followed with a very high level of scrutiny. You're not just doing chart reviews to say, oh, this person is prescribed hydroxyurea and azithromycin from 10 years ago and things like that. You're actually checking for the disease-modifying drugs. You're checking, like, did this person pick up their medicine, or did they get this dose of crizanlizumab? And then you use basically sort of back-end data techniques like propensity score matching and things to create what we call cloning or pseudo-randomization to figure out. So here are just the biomarkers that are the drugs and what we do know about the drugs. And basically, hydroxyurea is the only one we know anything about. And this is the sort of predicted consort diagram of how these thousands of patients will probably flow through the study. And so we're collecting all this data. And the additional thing that we decided to do is DNA. So we're going to collect DNA from patients once during the study to do whole exome sequencing. And we'll look at all these markers. We'll look at all the stuff that they would look at in the clinical trials, like how many pain crises, but we're also looking at organ injury, which I think is very important. And just some preliminary looks at our own data when we dove really deep into our own data and checked, for patients who are truly taking medicine A versus medicine B, and we have a period of time we can analyze where we're 100% sure the patient actually got the drug, and we have biomarkers before and biomarkers after. We actually see some trends here that crizanlizumab looks like a better drug. In terms of proBNP, if you look at the delta for patients who started voxelotor versus crizanlizumab, it's a small group of patients, about 20 in each group or 25 in each group. You're seeing a delta for criz, and you're not seeing a delta for proBNP. Same for urine albumin to creatinine ratio. We see a pretty dramatic drop with people who start crizanlizumab. And then voxelotor for hemolysis, this one really surprised me because this drug inhibits hemolysis. But what we see is that it seems to really be driven by super responders here. That, we have some patients who will take the drug and shoot from 7 to 12, but there's a lot of non-response in voxelotor, and we are seeing that clinically. We're also going to do some secondary analysis of breaking out the different sub-phenotypes of sickle cell disease, high hemolyzers, more vaso-occlusive phenotypes, et cetera. And this is just more of that voxelotor data with the super response. So, we have some people who just basically have these huge responses. So we'll look at that genetically as well. And we plan to do some deep dives on those patients, looking at single cell RNA-seq and the transcriptome, et cetera. So that study, everyone's IRB approved. We should be probably launching in the next few weeks. And so call me in 5 years and I will let you know which drugs are the best. But for the present, we have this problem of we have drugs. We don't really have a good way to measure sickle cell disease. We have tons of labs that we kind of follow. We watch them go up. We watch them go down. It's not ideal. And so another project that we are actively in is trying to measure sickle cell disease severity and response to treatment through the eye, because the eye is a transparent window to a disease-sensitive vascular bed called the retina. So what we had discovered is that if when you go to your retina doctor, you probably get OCTA done, which is just a picture of the blood vessels in the back of your eye. But we realized that if we take multiple pictures from an OCTA machine and then layer them on top of each other with the image processing algorithm, we can identify blood vessels that have disappeared and then reappeared and quantify how often that happens. And those episodes where a blood vessel is there and then 2 minutes later it's not, that's a vaso-occlusive episode, right? There's a vaso-occlusive event, and we actually have some microscope data where we can show it happening. So what we created was a sort of a metric of how often this happens, and it just comes out as a number, basically the percent of the retina where we're seeing these types of events. And you can say this is a patient before and after hydroxyurea. Oops. And those flickering blood vessels, you see less of them coming in and out on the post-treatment. And so this metric, we had a few different ones, but the one we're using is the one on the left. We call it intermittent flow index, or intermittent perfusion index. And you can see that people without sickle cell disease, they don't have this. It's the people with sickle cell disease are having these events. And so, and then when we sort of looked at this intermittent flow index across the different types of sickle cell disease, it really seems to correlate very nicely with your disease severity. So this is hydroxyurea, and you can see the computer basically highlights these episodes. Red is where a vessel disappeared, blue is where a vessel that wasn't there suddenly appeared, and then we quantify that. And you can see the patient before hydrea, 2% of their retina had this, and after 0.74. Well, what about when you cure people with gene therapy? What do you get? Well, so this patient had an intermittent flow index of 2.83. And that was after an exchange transfusion, so probably more than 50% of his blood was hemoglobin A at that time. He was in the CRISPR trial. After a second exchange transfusion, his intermittent flow index went down to 0.73. After the second exchange, you're probably 85% normal blood. And we think of exchange transfusion as, I wouldn't say it's a cure, but right after an exchange, most of your blood is, you have less sickle blood than a patient with sickle cell trait. So it is as close as we can get with a treatment to making somebody not have sickle cell disease. Then he got your CRISPR Therapeutics gene editing, and his post-gene therapy fetal hemoglobin is around 50%. And then you can see his intermittent flow index stayed right where it was after 2 exchange transfusions, and it's been like that since. And this patient does not have pain crises anymore, and all his organ function has completely normalized. He has some residual bone injury from arthritis he accumulated. We also, to understand what is actually happening when these vessels are disappearing, we have something called an adaptive optics microscope, where we can actually take microscopic videos of the cells, and we are seeing different types of things. And so this is a video of a sickled red cell. This is a little bit of a clump. Actually, you can see a few cells in there occluding blood flow. And so we've identified a few different mechanisms for the way that blood gets stuck in blood vessels, but we have confirmed that it's red cells that are basically not moving in the vasculature. Interestingly, it's usually not white cells, which some of our mouse models, seems like a bigger role for leukocytes. So that is that project. How am I doing on time? Perfect. So this is all great, but sickle cell disease is a genetic disease that is very amenable to potentially being cured with gene therapy. And so that is, I think what we would like the most would be to put ourselves out of business and have this just be a disease that you may be born with, but you'll be cured, and that's that, and it's really not much of a thing. So there are a few different ways that gene therapy is being used to cure people with sickle cell disease. I'll go over a little bit with the fetal hemoglobin approaches. That's the CRISPR Therapeutics is using that one, but basically, when you're a fetus, you're making fetal hemoglobin, which is it's transcribed from proteins, genes that are at different spots on the genome than adult hemoglobin. That fetal hemoglobin does not have a mutation. Your mutation is in your adult hemoglobin genes, and so if you can get your body to just decide that it would make fetal hemoglobin instead of adult hemoglobin, you could theoretically be cured. And the more, the better. We know that there are certain genetic abnormalities where people make 100% fetal hemoglobin, and it is a completely benign condition. There's nothing that fetal hemoglobin can't do. It doesn't even cause problems during pregnancy. So the more, the better. And you can see that basically there's all these transcription factors that kind of get together in different ways to regulate that switch from fetal hemoglobin to adult hemoglobin. BCL11A is a big one. So how do we do this? Well, the two big companies on the market coming to FDA approval are CRISPR Therapeutics and Bluebird Bio. And Bluebird Bio uses a lentivirus. And so basically what they do is they are inserting a transgene. So the lentivirus carries an entire gene for a normal hemoglobin, and it will invade the cell and drop that gene somewhere. It's actually relatively random where it goes. There's no specified place, but by and large, when that is done, it's completely benign, and then you start transcribing this normal hemoglobin, and it's actually a tagged hemoglobin, so they know how much of it you're making. It's called 787Q. It's not just normal adult hemoglobin. It's a little bit more similar to fetal. It has a higher oxygen affinity, which is a good thing because that prevents sickling. CRISPR Therapeutics is using CRISPRs. They're editing a spot on BCL11A called +58, and basically BCL11A is a gene that turns off fetal hemoglobin. So if you turn off BCL11A, you effectively turn on fetal hemoglobin production. How is this actually done? Well, you bring the patient into the hospital to an apheresis unit. You put a large line in. You give them a stem cell mobilizing agent, not GCSF, because that makes people with sickle cell very sick. Plerixafor is what we use. And you harvest as many hematopoietic stem cells as you can, CD34 positive stem cells. Those are taken to a facility, to a manufacturing facility where either the CRISPR special sauce is added or the Bluebird Bio special sauce is added, and those cells are edited, and then they are expanded using proprietary cell expansion protocols until you have basically a product that has around 500 million stem cells that are going to make blood that is not sickle cell blood. It's still the patient's blood, but it has been edited. Then you myeloablate the patient. You bring them to a bone marrow transplant unit. Right now, all these protocols are using busulfan, which is a very toxic myeloablative agent. There's radiation as well. And it's actually very important that you do a pretty complete myeloablation. You're not, even though the patient doesn't have cancer, there's two things. First of all, you want to kill as many cells that are going to make sickle blood as possible. And also, the partial myeloablation trials where they gave lower doses of busulfan, you basically had injured cells with DNA damage that survived. And so there was more issues with potential malignancies and such. So the myeloablation is complete, which means that the patient will be very sick, very vulnerable. They'll be in a bone marrow transplant unit for a month, month and a half. But you receive this myeloablation, and then we basically hang a bag of this product, which has come back from this manufacturing facility. Those cells will go into your body because you've opened the bone marrow niche by giving busulfan. The patient will slowly start to make their own blood, but blood that does not have sickle cell disease. These are the results from the Bluebird Bio trial. CRISPRs look pretty much exactly the same. Basically, once you've had the therapy, you stop having complications of sickle cell disease. There is some like lead-in period. Some patients with chronic pain may continue to have certain issues, but by and large, you stop having complications. I've had a couple people who've had Bluebird Bio. Their hemoglobins are above 13.5. You get about 8 milligrams of this 787Q hemoglobin from the treatment. CRISPR therapeutics, the fetal hemoglobin levels are not quite as high as Bluebird Bio. So if you check a fractionation of a Bluebird Bio patient, it's going to be 75% 787Q, this gene-edited hemoglobin. CRISPR therapeutics patients, they make about 50%, 40 to 50% fetal hemoglobin, but they're both cures. So you're above this kind of threshold where it's basically a curative treatment. Problem is that it is not a cure for everybody. First of all, 90% of patients or more probably would not opt for this because there's a risk that it can kill you. That risk is probably around 1 to 2% just from the it's not the gene therapy, it's the busulfan and the how sick you'll be after the myeloablation, and then there's a secondary cancer risk with the busulfan. Most patients don't want this. Most patients are not fully ready to risk their life to be cured. And most patients want to have children, so that's another major barrier. So what's the real, holy grail for sickle cell disease? In vivo gene therapy, which would be a product that you inject into the patient and it goes and edits the cells that make blood in the patient rather than having them taken out. This is a lot tougher because when you're doing it outside the body in a dish, you have the advantage of, first of all, you have 100% cell selectivity. You don't have to have some kind of targeted lentivirus or anything like that because you've selected the cells before you administer the therapy to them. And number two, you have the ability to sort of check what you did, right? If you edit these cells in the dish, then you expand them, you can sequence them to check, did I insert that lentivirus into any tumor suppressor genes or anything like that? And if something goes wrong, well, you just don't have to give it back to the patient. Anything you do in vivo, if it's something that you regret, well, it's in the wild and you're done. So we very much need improvements. So our research, we are looking for a gene, this is Jim Beaker and me, looking for a gene that has some selectivity intrinsic to it. So one of the problems with BCL11A, which is what CRISPR Therapeutics uses, that gene is expressed in leukocytes, that gene is expressed in the brain, that gene is expressed everywhere. And so you would have, it's a proto-oncogene, you would have concerns about editing a gene like that in the body without having a product that could really home just to the cells that you want. In fact, you don't even want to get to stem cells, you want to try to get to cells that are making red blood cells, which is a tremendous challenge. Whereas KLF1 is a gene that has similar functions. It's a regulator of erythropoiesis, but it is expressed exclusively in red cells. So if you were to edit KLF1, you wouldn't have, in cells that were not going to be red cells, you would not have such great concerns. And part of the reason we know this is that there are human beings who have mutations to KLF1 where they are insufficient in it, and these are benign mutations, but these people make high levels of fetal hemoglobin. So we tried to find an edit that, where can we edit KLF1 that would cause it to mimic what these populations in Malta and Asia have, where it's a benign condition, but they make a ton of fetal hemoglobin. So the first place we went after was called intron 1. And the reason we targeted this site is because there's a very highly conserved region. If you follow that red A down across all these different species, everyone except for the dogs shared this base. And so we thought, well, maybe this is important. Let's see what happens when we cut it out. And so one of the things about CRISPR is just because you have the sequence, you have the guide, very often you still just don't get editing. And so we had to tweak the system quite a bit, and we wound up with basically using 2 different guides to come at different, from both sides to try to chop out this one base. And using that approach, we were finally able to get very good deletions of that area, greater than 99%. And when we looked in cell lines, we were seeing reductions in KLF expression. So we were thinking, okay, well, KLF is a gene that if we turn it partially off, it'll be like these people who are haploinsufficient, and they'll make more fetal hemoglobin. It worked actually in leukemia cell lines, these like immortalized cell lines, but when we went to human CD34 hematopoietic stem cells, we made the edit, and it basically did not change the behavior of the cell. Just continued to make, maybe 20% more fetal hemoglobin, but definitely not something that would be curative. So that was 3 years and a disappointing result. But then we went after a different spot called EHS1, which is another regulator region of the KLF gene, where if you were to take out the whole EHS1, you'd have like a 100-fold drop in expression. And that is actually not what you want. So one of the big challenges with KLF1 is if you're completely insufficient in it, that is, you're going to have a cell that basically doesn't produce blood. So you need a way to sort of turn it down. So what we started doing is saying, all right, well, EHS1 is a very potent enhancer. Maybe if we just try to take out a small piece. So we designed a bunch of CRISPR guides to try to take out small pieces of EHS1. And we did find one where our RNA expression was where we wanted, you know, the 30, 40% drop in KLF expression. So, okay, now let's check this one to see if are we seeing increases in fetal hemoglobin. So you can see over here, control cells are cells that are just edited with scramble CRISPR. Our positive control is the BCL11A edit that is in Vertex Therapeutics, and that raises fetal hemoglobin about 4 times. Our EHS1 edit alone raises fetal hemoglobin 7-fold, and then when we actually combine it with the original intron 1 edit that we've been trying for several years, it raises fetal hemoglobin about 20-fold. So this was very exciting. We're super happy. We now have a target that you can edit. And the beauty of this target is that if you were to send in some kind of gene editing vector and you wind up editing the liver or the brain or, you know, off target, basically everything except you definitely don't want to go into the germline, this could work, and it would be much more likely than BCL11A to be safe. So how do you do that? Well, lipid nanoparticles is the approach that we're pursuing at this point. You got viruses. So viruses is one way to do it, and nanoparticles is the other predominant approach that you can take. Viruses have the benefit of they have specificity, so you can sort of send them to home to certain things. The downside is that you can have immune response to viruses. Generally, these are treatments you can only get once with a virus, and then you have formed an immune response, and that's it. So lipid nanoparticles, you can just give them over and over again. They have a variety of formulations that are non-immunogenic, so it's something that can be given. The challenge that we have come across is that, well, there's a few things. First of all, lipid nanoparticles, we know they're fantastic at delivering messenger RNA. That's basically your COVID vaccine is a lipid nanoparticle with a messenger RNA. They're not as good at delivering gene editing messenger RNA because a lot more needs to happen, right? You're sending in an RNA that's going to be translated into a CRISPR protein. That protein has to hook up with a guide RNA that has been sitting in that cell. That's about a 24-hour process, and then that combined guide RNA and CRISPR protein, which is called an RNP, then has to go into the nucleus and edit. So in situations where you're seeing 80% expression of just RNA, you may see 10 or 15% expression with your gene editing. The other issue is that these LNPs are not cell-specific, and lipid nanoparticles, when you inject them into patients, they tend to wind up 98% in fenestrated organs, meaning liver and spleen. So if you have a disease that is, like hemophilia or transthyretin amyloidosis, where what you need to edit is in the liver, that's great. But when we need to edit the bone marrow. So what we are looking at is 2 approaches. One is to take a nanoparticle that's, just a garden variety of nanoparticle, but inject it directly into the bone. And for me, as an emergency physician, I'm very comfortable with this. The hematologists who do bone marrow biopsies are also probably quite comfortable with this. We put intraosseous lines in the ER all the time when we cannot get access. And so we are exploring that. And the advantage of that is that these lipid nanoparticles are really today's technology. We can make them, we can manufacture them, we can turn them into a drug. And so if the key is just injected directly into the bone, then you really are very close to a drug that could be put in a box, sent wherever, including Africa, and just administered to people via intraosseous injection. The alternative is homing nanoparticles, which are, it's a little bit more of a future proposition. We have them today. You can acquire them and do your research and get your Nature paper, no problem. But in terms of manufacturing them, and being able to GMP the product, that's not quite there yet. So we're hoping that we can get this working with a non-antibody conjugated nanoparticle, so that we have less of a hurdle on our way to hopefully having a gene therapy cure. So that is that, and I'll be happy to take any questions. Thank you. Mm-hmm. Yeah, so I've done a lot of adaptive design stuff. We all love Bayesian adaptive trials. The difference here is that this is not actually a trial at all. It is actually, it's a extremely fancy cohort study. It's just an observational study. Well, if you were going to do an adaptive design, that would mean you are assigning patients to treatments. We are not. We are just letting doctors prescribe and whatever regimens the patients are on. And one of the cool things is that we find that patients wind up on multiple different regimens, sometimes because of indication bias, but sometimes because of access and insurance cut this patient off, and we'll get these natural sort of crossover events because patients are being denied care by their insurers and things like that, which is a horrible thing, but we hope to take advantage of that. But yeah, so because we're not assigning anything, we're just using data on the back end. I'm definitely going to try some Bayesian statistics, because what we found is Bayesian statistics is very powerful for small, rare diseases, things where you have sparse cells in your mega regression and things are just not converging. You can still get estimates of what does this mean when you're using Bayesian statistics.
So, Jeff, the field seems kind of ripe for evaluating combinations. Mm-hmm. Driving rational combination opinion adaptive biomarker type strategies. Are these drugs used? How do clinicians select which drug to use based on what's the cost of these drugs?
Yeah. Well, I'll do the cost first. The voxelotor is $125,000 a year, and crizanlizumab is $80,000 a year, and hydroxia is, I don't know, it's 25 cents a month or something like that. And Dari, which is this protein powder, is $30,000 a year. So they are used in combination by people like me and the other people at these centers whenever we can. The way we get people on combination therapies is you get somebody approved for one, and then you get the insurance company, the drug company, to give you free medication for a year on the other. And Novartis and Pfizer are both doing that for their drugs. I think they both think they have the best drug, and so they're hoping that patient A will go on crizanlizumab when they're already on voxelotor, and when their year is up, they'll decide, I'll have my insurer, because the insurers will pay for one drug in addition to hydroxyurea. So we'll have a lot of combo data there. But the events where you have somebody on 3 or 4 are usually based on free medication, or somehow patient just like coasts through. I like can never understand why it happens, but sometimes we prescribe a third $100,000 medicine and they just approve it. So we have enough of those events that we do think we'll be able to look at these combinations. And I think if there's a signal there, then maybe we would pursue some kind of trial where it's actually an interventional trial where assigning patients to things, and then Bayesian adaptive designs would be. I think that type of design is crucial for rare disease research. Yeah.
Jeff, thanks for the great talk. Obviously, a lot of the risk of the bone marrow treatment, which sounds very effective, is ablating your bone marrow. It seems very not indiscriminate.
Yeah.
Is there any potential work out there with just ablating the erythroid line?
Yeah. So I think the intermediate thing between these in vivo gene therapies where you're just injecting, there are antibodies, right, that can ablate CD34 cells without all this like toxicity of busulfan. And there's a few companies that are basically their myeloablation regimen is an antibody. So those will be probably over the next few years. And I do think that those will replace Bluebird Bio and Vertex. I'm not sure if Bluebird Bio and Vertex are doing an antibody-based trial, but they should, because certainly once those are available, everyone is going to want to be, up and running with a gene therapy that uses an antibody because it's less toxic.
Great talk.
Thank you.
What showed a signal of harm with ICS related cancer?
Yes.
I was just wondering if you had any comments.
So first of all, how did I get a good result on the first trial and then basically show the opposite in the second? So the first trial, we did not have an enrollment criteria that you were on a stable dose of hydroxyurea. And this was earlier in my career when basically every week I was seeing half new patients, half follow-ups. And I think a lot of patients in the original trial were getting titrated up on hydroxyurea, and it was not an enrollment criteria that you have to be stable first. So I think that some of the benefit of inhaled steroids in the first trial was due to confounding with hydroxyurea titrations. We know that steroids in systemic steroids in sickle cell disease, the minute you stop, it precipitates crisis. It raises your leukocyte count. Our hope was that if you decrease inflammation in the lungs, that is the organ that reverses sickling and oxygenates the blood, and that with better oxygenation you would have lower complications, and that the dose would be low enough that you wouldn't have these other issues with steroids. What I think you're seeing here is the real harm signal is with acute chest syndrome, which is essentially people showing up with infiltrates on their chest x-ray. So probably turned out that whatever beneficial effect we had from inhaled steroids was trivial, if not nothing, and then people were getting more pneumonia. And so that's where you see this acute chest syndrome signal.
Question in the chat. You mentioned cost of those drugs. So the question is, could you address the role of racism in the time to hydroxyurea approval and use?
Yeah. So I think that what you're dealing with is the fact that if there's not a pharmaceutical company that stands to make several $1 billion on the approval of a drug, the ability of the sickle cell community to advance that forward is vastly inferior to the pharmaceutical industry. There was no money in it for hydroxyurea. This was an patent drug, and it was really left up to the community of researchers, which historically has way less funding. Things are getting better now, but we're always compared to cystic fibrosis, right? Cystic fibrosis has the CF Foundation, is a multi-billion-dollar organization. They fund centers, they fund research, they have their own patents. Our foundations have no money. And so, especially foundation funding and federal funding, we were underfunded, and our ability to move a drug like hydroxyurea forward was, trivial in comparison to what CF and the CF Foundation could do for its patients. And that is certainly a form of structural racism, right? It's a health disparity. But with these newer drugs, we have found that, pharmaceutical companies have realized that with a biotech startup, you can have a business model where you will make a tremendous amount of money, right? Glycomimetics was crizanlizumab before it got bought out by Novartis. And so, we're seeing that in lots of rare diseases, that there are ways for biotech startups to make money. And so that has been an improvement in the situation.
Let's say Dr.